Hello students, welcome to today's lesson on classification of bacteria. In this lesson, we are going to study classification of bacteria based on two biological features. These two biological features are shapes and interaction of Gram's stain with cell walls of bacteria peptidoglycan. Let us start the lesson by defining classification. Classification is a technique to categorize organisms based on their biological features. In this lesson, we are going to classify bacteria, one based on shapes. Since bacteria have different shapes and arrangements, they can be grouped under three major groups. These groups are 1. Cocci. Bacteria that belongs under cocci exhibit spherical shapes like Escherichia coli, Lactobacillus, Salmonella typhi, and Clostridium tetani. The second group, bacilli. Bacteria that belong under bacilli or bacillus have rod shapes like Streptococcus pyogenes, Streptococcus thermophilus, Streptococcus lactis, and Staphylococcus aureus. The third group, spirochetes. Bacteria that belong under spirochetes exhibit spiral or corkscrew shapes like Treponema pallidum, Borrelia burgdorferi, and Vibrio cholera. Here are the diagrams of shapes of bacteria. The first groups of bacteria exhibit spherical shapes. The second group exhibits rod shapes. The third group exhibit spiral or corkscrew shapes. When we see their existence in their own habitats, some may exist or arrange themselves in single, some might exist in pair forms, some may exist in chain forms, and even some may exist in cluster or in colony forms in their own habitat. The second biological feature to categorize bacteria is based on Gram's stain. Based on Gram's stain, bacteria can be grouped as gram-positive and gram-negative. First, I think we need to explain what's Gram staining. Gram's staining or differential stain is a test or a technique that uses staining to classify organisms, in this instance bacteria. So, based on Gram's staining, bacteria can be gram-positive. Gram-positive bacteria are a group of bacteria that always stained purple by Gram's stain. These include Clostridium tetani and Mycobacterium tuberculosis. The second group is gram-negative bacteria. Bacteria that grouped under gram-negative always stained pink by Gram's stain. Example, Neisseria gonorrhea and Vibrio cholera. This technique developed by Hans Christian Gram in 1884. Here is the structure of bacteria. Gram-positive bacteria have thick peptidoglycan, but gram-negative bacteria have less or thin peptidoglycan, and this thin peptidoglycan simply covered by outer membrane or capsule. Procedures of Gram staining. How we can differentiate either a given bacteria are gram-positive or gram-negative. Here is technical procedures to differentiate either a given bacteria are gram-positive or gram-negative. First, a clean, grease-free, dry glass slide must be prepared. Second, a smear of suspension should be added on a clean, dry glass slide by a wire loop. Third, slide cells must be air-dried and heat-fixed. Fourth, crystal violet should be poured and kept for about 30 seconds to 1 minute and rinsed with water. Fifth, Gram's iodine should be added for 1 minute and then washed with water. 6. Slide cells should be washed by 95% concentrated ethanol alcohol or acetone for about 10 to 20 seconds and then rinsed with water. 7. A counter stain or safaranin should be added for about 1 minute and washed with water. And lastly, slide cells should be air dried through blotting and observed under microscope. After all these procedures gets performed or accomplished, bacteria might exist purple or red in color. You see, when bacteria is stained with primary stain crystal violet and fixed by the mordant, some of the bacterium are able to retain the primary stain and some are decolorized by alcohol. This is due to the cell walls of gram-positive and gram-negative. The cell walls of gram-positive bacteria have thick layer of protein-sugar complex with some lipids called peptidoglycan. So, decolorizing the cell causes this thick cell wall to dehydrate and shrink, which closes the pores in the cell wall and prevent the stain from exciting the cell. So, the ethanol cannot remove the crystal violet iodine complex that is bound to the thick layer of peptidoglycan of gram-positive bacteria. Then the bacteria appear purple in color. But in case of gram-negative bacteria, cell wall also takes up crystal violet iodine complex, but due to the thin layer of peptidoglycan and thick outer layer which is formed of lipids, crystal violet iodine complex gets washed off when they are exposed to alcohol, ethanol as a decolorizer that dissolves the lipids in the cell walls, which allows crystal violet iodine complex to leach out of the cells. Then, when stained with safaranin, they take the stain and appears red or pink. In a sense, it means they retain colors of counter stain or safaranin. Here is the diagrammatic representation of the technical procedures. First, the sample of bacteria must be fixed on dry glass slide. Second, crystal violet should be poured. Third, iodine should be added on slide cells to fix crystal violet with peptidoglycan of the cell. Fourth, 95% concentrated ethanol alcohol must be added, or the slide cells must be washed by 95% concentrated ethanol alcohol or acetone. Fifth, slide cells should be stained by counter stain or safaranin. And lastly, if a given bacterium is gram-positive, they stain purple. If a given bacterium is gram-negative, they stained pink due to the above reasons what I have mentioned before. Let me invite you an animation that shows how we can differentiate sample of bacteria in laboratory.
The gram stain is the most widely employed staining method in microbiology. It is a differential stain because it divides bacteria into two classes, gram-positive and gram-negative. In the first step of the gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, The slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of Graham's iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick, dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the crystal violet iodine complex.
Comparison of gram-positive and gram-negative bacteria. As we mentioned before, gram-negative bacteria have much less peptidoglycan in their cell walls as compared with gram-positive bacteria, since gram-positive bacteria have much thick peptidoglycan in their cell walls. Second, gram-negative bacteria have membrane outside peptidoglycan, but gram-positive bacteria have no membrane outside peptidoglycan. Third, the outer membrane of gram-negative bacteria secretes endotoxins. Due to this reason, gram-negative bacteria are quite resistant to many antibiotics. But since gram-positive bacteria have no outer membrane or capsule, diseases caused by gram-positive bacteria are less difficult to treat. They always cause less serious disease, but there is an exception, like tuberculosis. Mycobacterium tuberculosis that causes tuberculosis results in a more serious disease, but disease caused by gram-negative bacteria more difficult to treat. They always known to cause more serious disease. This is all over what I have on classification of bacteria based on shapes and the interaction of gram stain with peptidoglycan.